We define gene expression signatures as patterns of expression from genes that have related or similar functions, or a group of genes that have changes in coordinated changes in their expression in response to a specific stimulus, for example, or under certain condition. So these signatures may obtain from your own data, for example, when you perform differential expression analysis, or you can use some publicly available signatures such as those available through MSigDB. When you look at the literature, there are several gene set testing approaches available, and there are a number of Bioconductor packages that you can use to apply these gene set testings. For example, we have Lima, which implements several gene set testing functions. For example, we have Camera or Fry, but these are all in the context of differential expression. We also have Ensemble of gene set enrichment analysis that uses a variety of gene set enrichment algorithms in the literature to calculate the collective significance score for each gene set. We also have GSVA package that implements 4 different gene set enrichment analysis methods. Today, I'm going to go to the details of these 4 methods a little bit more because I'm going to be focusing on these methods in my talk. So we have ssGSEA. Before anything else, I should highlight that one of the differences that these 4 methods have with other methods, for example, those provided from Lima, is that these methods provide unsupervised single sample approach. And by unsupervised, I mean that they don't need to know about the phenotype labeling, and they can give you a score for each single sample. So ssGSEA compares the empirical cumulative distribution function of the genes inside of the signature with those outside of the signature using Kolmogorov-Esmirnov-like tests, and then normalizes the scores across all of the samples. GSVA, however, starts with kernel estimation of gene expression distribution across all the samples, and then similar to ssGSEA, it performs a KS-like statistic. PLAGE and Z-score start with standardizing gene expression across all the samples first, and then PLAGE performs singular value decomposition to calculate the scores, whereas in Z-score we have combining different Z-score for different genes in each sample, and then we have the scores. What's important here is that when we have a closer look at all of these methods, they all take information from all the samples in the dataset in order to be able to give you the score for each individual sample. So it means that if we change the composition of the samples in the dataset, a score related to an individual sample may actually change. So that was a motivation for us to come with the Singscore method, which is truly single sample and doesn't take information from any other sample in the dataset in order to calculate single sample scores. So what you see here is that if we have a directed gene set, and by directed I mean, for example, we know the direction whether it's upregulated or it's downregulated gene set, or we have signature that has both up and down gene set, then we can have a single sample. So this is a gene expression profile from a single sample. Then we can rank genes in increasing order for the upset and in decreasing order for the downset, and then we calculate the mean rank of the up and down separately, which is this step here, and then we normalize the scores according to the theoretical minimum maximum mean ranks, which are these two, which are obtained actually using a very simple arithmetic sum. Then we sum the up and down scores together, and then we center the scores around 0. But what if we have an undirected gene set? So when we don't know the direction of changes in gene expression, what we do is slightly different. We median center the ranks from here. So the higher the rank as we go further from this median centered rank, and then we take the average of these ranks again, and similar to what we had for the directed gene set, we normalize according to the theoretical minimum maximum scores, and the scores range between 0 and 1. In my talk today, I'm going to focus on the situations where we know the direction of the gene sets. So the first scenario. But before anything else, we wanted to compare our method to the other available method to see how well we perform. The very first thing that we wanted to focus on was what happens if we change the composition of the samples in the data set, whether it changes a single sample score or not. And to do this, we looked at the TCGA breast cancer data with 2 platforms. We have RNA-seq and macroarray. And there are 500 samples that are actually overlapping between these 2 platforms. So these are the same patients, but they have their gene expression profile using both platforms. So if we focus on 500 overlapping samples, then for each single sample, we change the composition of the samples in the data, and we also change the number of samples in the data, and we further change the number of genes measured in the data set. Then you can imagine that for this single sample, we score that single sample using all the available scoring method, and then we go to the next sample, and we do that for 500 samples that we have. Then we will end up with 500 pairs of scores, and we calculate the Spearman correlation and concordance index, which basically quantifies the amount of concordance that we have between each pairs of scores in RNA-seq with the corresponding pair in the macroarray data. And obviously, the higher these values are, the more stable the scores are. So what we've used is a mesenchymal signature, and then we change the number of samples from 2 to the full set of 500, and then we change the number of genes down here, and we also calculated, as I said, correlation and concordance index. I'm going to presenting different methods in different colors, and as you can see, we also included the ssGSEA without normalization. And the reason that we did this was that, if you remember, the ssGSEA method performed normalization at the very last step, and so we wanted to see if we skip the last step normalization, if the method performs well or not. And so here are the results. We have, for the full set of 500 samples, it doesn't really matter which method we use, they all perform equally well. But as we go further towards the smaller data set, for example, with 2 or 5 samples, we see that the stability of the scores obtained by other methods drops dramatically. And the only methods that perform still stable are the Singscore method and also the ssGSEA without normalization that we had. You can also see that PLAGE method also changes its stability if we change the number of genes measured in the data set. But why we care about stability? What does that mean actually? To show you the importance of stability, I'm going to take you very quickly through one practical example. So imagine a data set where we have 2 conditions. So we have TGF-beta treated samples in red, and we have control samples in blue. And we score them against mesenchymal signature, where we expect to see the TGF-beta treated sample to have higher score. As you can see here, they have higher score compared to the control samples. What we can do is that we can take a few samples out from this distribution and score them separately and try to overlay them back to this distribution to see if we can put them in the right place. So basically, as an example, we might take 2 control samples out, and then we score them, we overlay them back. Or we might get 2 control and 1 TGF-beta samples, we score them and we put them back. And we have different scenarios here, as you can see. To do that for all the method, this is what we get. For the Singscore and ssGSEA without normalization, if you follow this, you can see that we get really pretty good results. So we have, regardless of which of these scenarios or which composition of samples we have, we always put the scores in the right place. But whereas I'd like to highlight here a difference between these 2 methods. So although the ssGSEA without normalization performs as well as Singscore, because we removed the normalization step, which was at the very last step, the score that we have here are pretty large. So it's very difficult to interpret the scores. Whereas what we do here, if you remember, we do pair sample normalization. And so we consider the theoretical minimum and maximum, and the scores are around 0. And it's very easy and straightforward to interpret. For the other methods, however, when we look at the imbalanced sample composition, like when we have 2 control only or 2 TGF-beta, we see that the methods don't perform well. So they basically put the samples in the wrong category. So that's why it's important for us to try to have stable scores. In the next step, we wanted to compare the power of methods to see if they can differentiate biologically different groups. To do this, we simulated RNA-seq data with 30 samples and 1,000 genes. Then we defined 2 groups which have 30 differential expressed genes between them. Then we created gene sets with different signal-to-noise ratio, where we have 80% of the genes in the gene set to be differentially expressed, 50% or none of them. Then we repeat this process 100 times, and each time we also vary the log-fold change or effect size, and then we score samples using all the methods, and then we actually do t-tests on the obtained scores to see how well they perform in separating these 2 groups. And we calculate power and type 1 error, which is the false positive rate. As you can see, except for PLAGE, all the methods perform pretty equal like in both 50% DE genes and also when we have the high signal-to-noise ratio, and the error rate seems to be pretty similar across all methods. In another scenario, we wanted to also compare methods in terms of their ability to call true differential gene sets between two groups. So what we do here is that we again simulate RNA-seq data. This time we have 10,000 genes and we have 2,000 differentially expressed genes between the two groups. And similar to the previous scenario, we have also 3 gene sets, but this time we take 500 gene sets for each of these scenarios. And then we repeat this whole process, and in each simulation we score samples against all the gene sets. We again perform t-test, but this time we calculate F1 score, which takes into account both precision and recall. What you see here is that for the 50% differential expressed genes in the gene set, we see all method performs pretty poor, but when we have a stronger signal-to-noise ratio, SINGScore performs as well as GSVA and z-score, whereas it's slightly better than both ssGSEA methods, and it's much better than PLAGE. In some cases, it happens that we want to score the same sample against many gene sets, and so that's why the computation time matters. We randomly selected 10,000 gene sets from MSIC-DBs, and also we subsampled the TCGA breast cancer data to have a small set of 25 samples and a large set of 500 samples. And you can see that the computation time is pretty slow for both ssGSEA methods, and the SINGScore is as well as all the other methods down here. In this section, I'd like to actually take you through some of the functions and visualization that the SINGScore package provides. So scoring samples is actually very straightforward. We rank genes using this function, and then we say a simple score, and it's simply score the samples. Then we wanted to study each individual samples in more depth. So what we do is that we provide this plot rank density function where you can take a sample, for example, with high score, and then look at the rank of the up and down gene set in that single sample. So here, because we have a high scored samples, we see that the older rank of the up gene set are clustered towards higher value, whereas the rank for the down gene set tends to be lower. If we take an intermediate sample with intermediate score, we see that the rank of the up and down gene set are randomly distributed, and even when we look at the down score, we may see something anti-concordant because this is very low score. So for example, we have the rank of the down gene set towards higher value. So these sort of plots may remind you of some relatively similar plot provided, for example, in barcode plot or in enrichment plot. But I'd like to highlight the difference between the visualization that these methods provide with what we provide. In all these 2 methods that I've actually included here, they need a set of samples with different conditions in order to be able to rank things. So they work by looking at a set of samples. Here, the ranking is based on the log-fold change between 2 groups. And here, the ranking is according to the correlation of gene with a specific phenotype. But what we do is that we provide single sample visualization when we look at the rank of the genes within each individual sample. And I also forgot to mention that these can all be interactive, so basically you can hover over those rank and see which genes perform better than other genes in the data. We also provide functions to generate p-values for the observed score within each single sample. So under the null hypothesis that the upregulated gene set genes are not enriched within the high abundance genes, and also downregulated genes are not actually enriched with the low abundance transcript in our data, we can do permutation testing. And so how it works is that we randomly select gene sets with the same size as our gene set, and we score the same sample again and again, and we basically calculate these empirical p-values. So here is the null distribution for this specific sample, here is the observed score, and this is the threshold that we consider. So basically for this specific sample here, which has a low score, it's not enriched in our signature, whereas we have a significant score for this sample here. We also provide some visualization for you to look at, like for example, the two-dimensional landscape. When you have, for example, two different signatures and you want to look at the relative molecular phenotype of samples. We have plot score landscape function that you can use, for example, to score here what you see are ssGSEA breast cancer data scored against epithelial signature and mesenchymal signatures. What we can do is that we can also project a score landscape, which is basically scoring new set of samples coming from independent dataset and overlay them on top of the background distribution. So what we have in the background are patients, whereas what we have in the foreground are cell lines, and we can see that they have the same sort of distribution related to epithelialness or mesenchymalness. I've called it cell lines according to the different subtypes. Specifically, I have also these 3 cell lines coming from an independent data set compared to the other cell line that I have here. And what we can do is that we can monitor the changes in the molecular phenotypes of these samples. For example, we stimulated these specific cell line with TGF-beta and made them more mesenchymal, and you can see how it actually moves on this landscape. And then we applied or added some microRNA, try to partially reverse the whole process, and you can see how it changes. Another thing that we can do with this sort of plot is that we can have a more refined molecular classification of samples. Imagine we can take all these samples down here. Basically, not all of the luminal subtypes that we look at are here. There are some of them up here. And also not all of the claudin low subtypes of breast cancer are here. As you can see, some of them are here. So we can have a more refined classification of the already existing the classification out there. And then you might want to do some sort of analysis like differential expression analysis between these two groups, or look at other levels of data, how they are different in terms of methylation, in terms of the mutation, or any other omics. So in my talk, I introduced Singscore, which is a very simple, straightforward method. It's ranked based, and it's truly single sample, and it generates interpretable scores. I showed you how we can compare this method in terms of stability, power, F1 score, and also the computation time, and we showed that it either performs as well as other methods, and in some scenario, it even outperforms some of those methods, for example, in a small data set in terms of stability. And also, because we have this as an Bioconductor package, come to me to get the stickers, it is also reusable. We also have a Python version of this, which is still under development, but it can generate the basic functionality of this method. We have both static and interactive visualization, which are both at bulk level and also at single level. We think that this method can be used in preclinical or clinical research in order to actually to have more refined classification of samples and better understand the molecular phenotype. So this work has been supervised by Melissa and Joe from WeHi, and also Damesh had done a lot of work for comparing different methods, and Ruchin did a lot of work for developing the Bioconductor package. I'd like also to thank Terry and Gordon and all the funding resources. I'm happy to take any question. Thank you. Yeah, so we have. Would you mind just repeating the question? Yeah, so is that like, correct me if I'm wrong. So the question is, what are used to score these samples? So basically, if you look at the literature, there are epithelial and mesenchymal signatures available. So there are some signatures specific for tumors and some signatures are specific for cell lines. So basically what I've done here is that I've scored tumor samples in the background by using the signature which is specific for tumors. And so these are like about 300 genes, and the foreground is a little bit less genes, and they have some overlapping genes, but they are specific for mesenchymalness or epithelialness in cell lines.
Probably I missed some part. I understand how you rank the genes in one single sample.
How we rank according to their abundance, or how we rank genes in each single sample according to their abundance of the transcript.
So
Gene
it's
expression.
absolute abundance. Because you are measuring honesty. So that kind of thing you cannot get an absolute value of the abundance.
What do you mean by absolute mean without the actual sign? We do, for example, if you look transform, we might have values from -5 to say 15, right? So we rank from -5 to 15 according to the expression value in increasing order.
So the expression value is the recount number, or you transform that or transform?
That's a good question. What's the format of the gene expression? That's your question. Yeah, the input for that. So you actually do any normalization that you do. For example, if it's RNA-seq, you calculate RPKM or look to transformation of RPKM, and you further filter for the low expressed genes because we don't want to have like 2000 in our data. So that's just like a normal normalization that you usually do for transcriptomics data. This is after normalization. Yeah.
So you don't care.
We'll talk about that.